Hello everyone, this here is Lab 10, starting on page 111. We'll be talking about blood. Blood is a connective tissue, a liquid fluid connective tissue. There's three categories of cells: erythrocytes, which are all these little transparent pinkish blobs, also known as red blood cells; platelets, which are these teeny tiny little fragments; and leukocytes, or white blood cells. And there are many different kinds of leukocytes that you need to be able to identify. So, again, erythrocytes. Erythrocytes are the little blobs that are reddish with little clear spots a lot of times. Those are red blood cells. They carry oxygen. They are the most common blood cell, Platelets, teeny tiny little fragments. They're important in helping in forming clots. They are the smallest cells in the blood, so they're teeny tiny. We have, out of the leukocytes or white blood cells, we have the lymphocytes. Here is a lymphocyte. It helps with specific immunity to fight very specific infections. It has a very large nucleus that fills most of the cell, and it is the second most common type of leukocyte. We have the monocytes. There are two images here. Their nucleuses are pretty big. Sometimes they're shaped like a U, sometimes like a kidney bean, and they engage in phagocytosis. So when they get into tissues, and only in tissues, they will engulf pathogens to destroy them. There are neutrophils. Neutrophils are the most common leukocyte, the most common white blood cell. They have their nucleuses all blobbed around. They're kind of purplish in color. And they also engage in phagocytosis. They engulf pathogens, bacteria, viruses, and destroy them. And neutrophils function both in the bloodstream and in tissues. Basophils shown here, they have lots of dark blue, dark purple blobs everywhere, so many so you can't see the nucleus. And they're involved in increasing inflammation, which is useful for a while for infections, but they also can cause the allergic response, which is a hassle. And then eosinophils, shown here, they have sort of the reddish colored granules, and their nucleus is a bunch of little blobs everywhere. They help to limit inflammation, so they do the opposite of basophils. And also they can fight parasites, big things invading our body like worms. And once upon a time, everybody had worms. All right, Hematocrit, that is the volume of blood that is composed of red blood cells, percentage of red blood cells by volume. And our red blood cells should make up about 45% of our total blood volume. Anemia means reduced oxygen carrying capacity. This could be caused by a low hematocrit, so if there's less than 40% of the blood is red blood cells, and that's a problem. Could be caused by not enough hemoglobin in the red blood cells, or it could be because of low iron. It turns out iron is critical for hemoglobin to bind oxygen. You don't have enough iron, your hemoglobin will not function. Polycythemia is the opposite. It's too many erythrocytes, too many red blood cells, over 55%. If this happens, then the blood gets really, really thick and adversely affects blood pressure and heart function. Lymphocytes mount intense immune response against a specific pathogen. They fight one thing at a time. So, for instance, influenza or COVID-19. Basically, there are some different kinds of lymphocytes. You have helper T lymphocytes. They turn on the specific immune response. They activate it against that one pathogen. Cytotoxic T lymphocytes that destroy our own cells that are infected by the pathogen. So in the case of influenza, if you have cells in your respiratory tract infected with the virus, cytotoxic T lymphocytes will kill the cells infected by the virus. And B lymphocytes, when activated, turn into plasma cells and produce antibodies. Antibodies are proteins that then bind to the pathogen, so in this case to the influenza virus, often inactivating the virus, or at least making it easier for our neutrophils and other immune cells to find and destroy. Antigens are specific molecules on the surface of a cell. So bacteria have antigens, viruses have antigens, our own cells have antigens. They're just something on the surface, a shape. So in this case, it's a triangular shape. And if that shape does not belong in our bodies, then we get an immune response against that foreign or invading antigen. Antibodies are proteins that can bind to the antigen. So the antibody is made to bind to this invading antigen. When it binds to it, it can inactivate or destroy the pathogen, or at least make the pathogen easier to be recognized by our own immune cells. And agglutination is when antibodies causes a group of cells or viruses or bacteria to clump up. These clumps are easier to identify, but if they get too big, they can cause problems in the bloodstream. Here is a quick video about how lymphocytes turn into plasma cells to fight specific antigens. So an antibody is a Y-shaped protein. It has sticky ends that can bind to different antigens. So each plasma cell makes its own antibody. That antibody binds to a very specific antigen. And again, an antigen is just a structure on the surface of the pathogen, just something on the surface, a structure, a shape. Why do we care? Well, vaccines allow us to produce antibodies without actually having to get sick. Blood transfusions, because if you get the wrong kind of blood, so if you're of a certain type and you get the wrong type, that can cause an immune response that'll lead to death and organ transplant rejection. If you're getting an organ transplant, you need it to match so your immune response doesn't attack it. So antibodies on erythrocytes, they have structures on their surface. Some of these structures are called A, some are called B, giving us the AB blood types. So A has A, B has B, AB has both A and B, and O has neither. There's also plus or minus. Some of these could have pluses as well, additional shapes, or they could not have pluses. So minus means the plus doesn't exist, and they're on the surface of the cell. So for instance, dogs, we could say white dog furred with white fur have O, black dogs have A, brown dogs have B, and brown and black dogs would have A and B. So they would be representing the different blood types by the color of their fur. So again, blood types, you have a special structure on the surface of your cell or you do not have it. A type structure, B type structure, both A and B structures, or none of the above. So when you want to do a test for blood type, you take a blood sample and you add antibodies that'll bind to the A antigen and antibodies that'll bind to the B antigen. So for blood type A, you'd get clumping agglutination with the A antibodies because they bind to the A antigens. They do not clump up for B because you do not have any B antigens on the blood cells for someone who's type A. For type B, the opposite, you get agglutination with antibodies against the B antigen. You don't for antibodies against the A antigen because, again, someone with blood type B only has the B antigen. For AB, both agglutinate because you have both A and B antigens in your blood. And for O, neither would agglutinate because, again, you do not have the antigens. They are not present. Antibodies for A, B, O appear shortly after birth and automatically will cause agglutination. So you definitely have to make sure you know your blood type before, or at least the doctors know your blood type before you get a blood transfusion. Again, antibodies bind to multiple cells, leading to agglutination. Those clumps of red blood cells can then block blood vessels, leading to stroke or heart attack. We also have Rh, so plus or minus. If you're plus, then you have the Rh antigen. If you're minus, you do not have it. So dogs with long hair will be plus, dogs with short hair will be minus. So these dogs would be AB plus. They have brown and black fur, and they have long fur, so they have all three antigens. O minus would have none of the antigens. Sometimes Rh is also called D antigen. We're just going to call it Rh. So either Rh plus, you have it, Rh minus, you do not have it. So here's how the test works. You have wells, you put in the blood samples, you put in the antigens, and then you will stir it up and see if you get a reaction. So for instance, here we have a reaction in two chambers, the A chamber and the RH chamber. We do not have a reaction agglutination in the B chamber, so this blood type would be A positive, A positive. Here, look at this. What do you think it is? It's AB positive. Effects in all three chambers. Here's another one. Which type of blood type is it? It is B positive. You have a reaction in B and RH, but you do not have a reaction in the A chamber. And then this one, it is O positive. Nothing happened in A and B. You got agglutination in RH, so that means it is O positive. No A antigen, no B antigen, but you do have the RH antigen. You can donate to anyone who has the same antigens or more. So someone who's A positive can give to A positive people and AB positive people. You can receive from anyone who has the same antigens or less. So if you're A positive, you can receive blood from A plus, A minus, O plus, or O minus. You will not react to these blood types because they have the same antigens as you or less. Universal donors O positive, universal donors O negative. They can give blood to anybody. They do not have any antigens. Their blood cells are smooth as a baby's butt. They're awesome. Universal recipient is AB positive. They can receive blood from anybody because they have all three antigens. However, they can only give it to other AB positive people. This is just the ratio. Doesn't really matter, but if you can look at it if you want. So we have column D, it's O positive. We'll skip that, whatever. It's possible for a mother who is Rh negative to have produce antibodies to attack a fetus who is Rh positive. The way this can lead to severe anemia and other problems in the infant. So basically, if the mother is Rh negative, after the first child is Rh positive, she'll develop antibodies against the Rh antigen. Then, if she gets pregnant again with an Rh baby, then her antibodies will attack the second infant, the second fetus with the Rh antigens in its blood. To counter this, they give the mom antibodies against Rh factor to try to block it so that her immune system never sees it, and she never generates antibodies against Rh and won't cause any complications during pregnancy. Here's some practice questions. An emergency room patient has an automobile accident. He's unconscious. He is found to be B positive. Which of these would be the safest to administer? B positive. What would be the safest to administer? It is B minus. Same antigens or less. B, that's good. Minus, plus, that's fine because minus means absent. All right, hope you enjoyed that video. Be sure to do all the activities for this lab.